Okay, let's go back. Crap. Okay, so I don't actually know where it left off on the last one, but key thing here is the difference between the dog and the bacteria. We want to know which one is false about these cells. So bacterial cells lack membrane-bound organelles. That is true, so we can cross it off. C, bacterial cells are much smaller than the dog's skin cells. That is also true. We know prokaryotes are smaller than eukaryotes. C, the dog's skin cells do not have chloroplasts. So this brings in some plant and animal vocab, and I'm sorry we got a little ahead of ourselves on that one. But key thing, it's an animal cell. It does not have a cell wall. We're going to get more into that right now. So animal versus plant. We have our animal on the left and our plant on the right. Key things here for the differences is there are 3 things in a plant cell that are not in an animal cell. Those 3 things are our chloroplast, our vacuole, and our cell wall. There is no need for you to draw these diagrams in your notes. I would just make note that these are the 3 things that are only in plants, not in animals. Okay, so the genetic material of an animal cell is found in the what? So genetic materials are DNA. That is found in our nucleus, cytoplasm, ribosomes, or vacuole. This is a review. All right, it is our nucleus. That is the brain of our cell. It holds all of the DNA. Okay, use the diagram of the euglena to answer the question. Ben drew this diagram after viewing a euglena through a microscope. Which aspect of the euglena provides the best indication that it is a plant cell? So which thing about this diagram proves to us it is a plant cell? A, the euglena uses a flagellum to move. That doesn't really specify anything about a plant. B, the euglena uses chloroplasts to produce food. Plant cells are the only things with chloroplasts. That is how you know that is the best answer choice. They're the only thing with chloroplasts. Okay, key eukaryotic cell organelles. So I have the organelles listed, the main ones that are important. The key ones that I would definitely know the functions of are in the green box. So if you know the functions, the basic functions of the ones in the green box, you're going to be okay. The chloroplast, the vacuole, the cell wall at the bottom, as long as you know that those are in plants and not animals, you're going to be good. So as long as you just know that those 3 things are in plants, not animals, you'll be fine. The main focus we want is in this green box. So make sure you pause the video here. I'm going to keep going, but pause the video here and write down these basic functions if you don't have them yet. Okay, which of these functions is most like the brain of the cell? So our brain of the cell tells the cell everything what to do. That would be our nucleus. Please don't ever get this wrong. You'll make me cry. Nucleus is the brain of the cell. Okay, the picture shows 2 structures present within an animal cell. Which statement best distinguishes the functions of these structures? So key thing here, I know this is the endoplasmic reticulum, or the ER, and I know this is the Golgi. My key words that I think of when I think of this is proteins, and I think ER is where things are made. Golgi, key buzzword with Golgi is it packages things. You think Golgi, you think package. So, looking through our answer choices, what is the only one that explains that? It is D. Rough endoplasmic reticulum produces materials. We know these materials are proteins. And the Golgi apparatus packages and ships them. Packages is our key word. Okay, cellular transport. Getting to the end of our review. So we have active transport and passive transport. Active uses energy. Think if you're going to be active, you got to have some energy. Substances move from low concentration to high concentration. And our examples are protein transport. Passive, you don't need energy. It needs 0 energy. And substances move from high concentration to low concentration. Make sure you're writing this down in your notes. Examples are diffusion and osmosis. So, key thing we're going to focus on right now is osmosis. So we have these 3 different terms that get a little tricky. So I'm going to have you go ahead, pause where we're at. I'm not going to talk through them. Pause where we're at and make sure you just write down these key bullet points about all of these terms. Okay, a single-celled organism without food has reduced supply of cellular energy. Doesn't have as much cellular energy. How does this affect the ability of the cell to conduct active and passive transport? So if we know that the energy is reduced, it's going to affect the type of transport that uses energy. Which one is that? It would be C. Active transport is reduced, but passive transport is not affected. Active transport is the only one that uses energy. So if we get rid of energy in the cell, we're going to screw up the active transport. All right, and our last question. The graph to the left shows the relative concentrations of different ions inside and outside of an animal cell. So we see the inside of the cell are all the dark bars, and the outside of the cell are all of the light bars. So we're just looking at how many of these substances are inside the cell and outside the cell. So for example, Na, we see there are not a lot of them inside the cell because this bar is so small, but there are a lot of them outside of the small cell because this bar is huge. So which process is directly responsible for the net movement of K plus and Mg plus plus? So we're only looking at K plus and Mg plus plus into the animal cell. So we want to know as we're going from outside of the cell into the cell. So from the light to the dark, we would be going from low concentration to high concentration because we're going from a little amount to a lot amount. If we think back to our definitions, which type of transport goes from low concentration to high concentration? Is it electrophoresis? We haven't even talked about that, so no. Passive transport does not use energy. It's the opposite. It is active transport from low concentration, these little tiny bars outside of the cell, into the cell at the high concentration. We know it is active transport. Okay, that is the end of the cell crash course. So make sure you finish those individual questions in your packet and turn them into Miss McGrath to get some feedback on them. You guys are going to do great. 1 down, 7 more to go. Woo!